So these are the triple questions for the disease and defence topic. The graph shows the number of cases of AIDS between 1981 and 1999 in two groups of males. Those were homosexual and bisexual and injecting drug users, so people who typically during the 80s were likely to be exposed to HIV. Describe the trends in the graphs between 1981 and 1990. Over the years, both groups of men, incidence of AIDS increases until the 1990s. At 1990s, both incidence of AIDS start to decrease. There are more cases of AIDS in drug users than in homosexuals, even though they start at the same number. The reason I maybe wrote a long extended answer is because it was worth 3 marks. Okay, so to comment on the general increase, then general decrease, and which one maybe was slightly more increased than the others. State a reason for the decline in the number of cases of AIDS between 1990. More awareness of transmission, increased use of antiviral drugs to slow down the progression to AIDS. Increased use of condoms, giving HIV users needles, etc. Explain why a person who is HIV positive may not have AIDS. HIV is the virus that you contract. AIDS is the disease that it develops into. So people can be HIV positive for years and years before they develop AIDS. Explain why AIDS cannot be treated using antibiotics. AIDS is a virus. Antibiotics only treat bacterial infections. Suggest why it is not possible to develop a vaccine for AIDS. There are different strains, a little bit like the cold, different strains. Ineffective vaccine needs time. Many people who develop AIDS die from other conditions, normally non-fatal. Explain why these conditions become fatal for people with AIDS. People with AIDS have a limited immune system, a decreased antibodies. They don't often destroy pathogens, so foreign bodies that enter their body, and that means that pathogen numbers increase. So we might get the cold and be able to fight it. Someone who's unfortunately contracted AIDS might not be able to fight it. Their body might not be strong enough, strong enough to make the antibodies. A diagram here shows the vaccination process. Name the type of white blood cell that produces antibodies, the lymphocytes. Okay, describe the process that takes place between stages 2 and 3. Lymphocyte has reproduced, divided and make a copy. Some of these will then fight the flu virus and some of these will become memory cells. State the importance of stage 3 to protect the body from the flu virus, memory cells. So the next time you get infected with the pathogen that causes the flu, you will have a rapid response. New forms of the virus appear almost every year in the UK. Suggest why the government report recommends that the flu vaccine should be given every year, because different antigens, so different antibodies needed. You need to undergo this process every year. Name the English doctor who first used the vaccine to treat patients in the UK. Edward Jenner. After an injection, blood clots at the site of the wound. Suggest why it's important for blood to clot at the site of the wound. Prevents blood loss, prevents pathogens from entering, stops infection. So if we can stop the pathogens entering straight away, then we'll have a better response. So this is talking about antibodies, using monoclonal antibodies and how they work and using them in pregnancy tests. Okay, so it talks about how you have to have a line in the control window to know that it works, and then you would have an extra line then if it's positive. So which test strip shows a negative result? This one here. So it's got the control window, so it shows it's working, and there's no line in the results window. Monoclonal antibodies used for pregnancy testings. Give another use to treat certain cancers. Explain how it would show a positive result. So you just had to kind of explain how the monoclonal antibodies work. Urine passes through a reaction zone. HCG hormone binds to a mobile HCG antibody. HCG binds to the immobilized HCG antibodies in the results zone and the control zone, and the blue dye appears. This then goes on to look at drug testing. Give an example of a recreational drug that is addictive, heroin. Many people suffer from withdrawal symptoms when they become addicted to drugs. How does the action of the drug make the person become addicted to it? It alters the body's chemistry. Then it asks you to look at a table. Doctors investigate cannabis smoking of 1,500 young adults. The table shows the percentage of cannabis smokers who became depressed. From the table, give 2 conclusions that you can draw from the relationship between cannabis and depression. So the first one is that women are more likely, you can see the numbers are a lot higher, so women are more likely to be depressed than men. And then the more cannabis you smoke, the more likely you are to be depressed. You can look at less than 5 times once a week, and you can see how it increases if you're smoking every day. Next question then is looking at a large-scale drug trial. And it says, state 2 control variables in the investigation, no heart conditions at the start and no high low density lipoprotein levels. What would the placebo be in this investigation? A sugar tablet. The trial gave reliable results. Give 1 reason why. Large number of people tested. This kind of question comes up quite often. So if we're only testing 100 people, that isn't a large trial. It's not going to give you really reliable results. Also 50-50 split. The trial stopped 7 months early. Why? Give 1 reason. Overall results, unfair to carry on the trial, knowing that it would get really good results for many people. The manufacturer of Resultin paid for the trial, but however, no manufacturers took part. Suggest 1 reason why the manufacturers did not take part in the trial, to be biased to the results, so they had no idea what was going on. It reduces the risk of heart attacks. Use the data in the table to explain why. It lowers both types of cholesterol. Okay, lowers both types of cholesterol. Cholesterols have been known to clog arteries, which can cause heart attacks, and that's a topic that you did last year in triple biology in year 10. I think we talked about this in class, but I messed up the arrows, so use the pink arrows. So stage and the information. Tests on humans, including placebos, used to prove that the drug is effective on humans. Tests on humans using very small quantities of the drug. The first stage in the clinical trial of the drug. And then finally, tests on animals used to find if the drug is toxic. So this question then looks at talking about caffeine, daily dose in development of human Alzheimer's in human. If you read through the paragraph then, it doesn't actually talk anything really about humans. It's more about testing on mice and looking at their caffeine dose. So the headline for the passage is not justified. Explain why as fully as possible. Trial was too short. I think they only gave them to them for a couple of days. And it wasn't even humans. Investigation was not repeated. Mice not the same as humans, which is a large thing. We test on mice an awful lot, but they're nowhere near the same biochemistry as humans. So what responses they have will not be the same as what we would have. And it also improved their memory loss, but didn't delay it, which is the problem with Alzheimer's, is that you are eventually going to get memory loss. This question then, graph shows the number of people with measles in the UK between 1940 and 2010. Compare how effective introducing the measles vaccine was with introducing the MMR vaccine. So you can see here's the measles introduction, here's the MMR. Use data from the graph. So both lead to a reduction. So you can see here, it's up here, and it's when both of them are introduced, it does actually show a rapid decrease. Measles vaccine caused a large drop. MMR wipes out the measles. If you see here, by the 2000s, there were no cases of measles. The MMR vaccine was introduced in 1980.